modulatory effects.
Thank you, Dr. Sylvester, for your kind introduction. Good morning, ladies and gentlemen. Today, my presentation is entitled Tocotrienols, Anti-Cancer and Immune Modulatory Effects. I will start my presentation with a brief introduction, and I will enter a little bit on the research approach, basically on a cell-based system using syngeneic mouse model of breast cancer, as well as some studies carried out on healthy human volunteers. After which I will go through the results and a discussion with some conclusions. Breast cancer, as we know, is the second most common cancer in the world after lung cancer. And the WHO report in 2006 has shown that breast cancer is the fifth most common cancer cause of cancer death. And in Malaysia, as well as in worldwide, the most common female malignancy is due to breast cancer. The National Cancer Registry in 2003 has shown that the highest percentage of cancer is caused by breast cancer in Malaysian women. And some of the cancer-causing factors include age, sex, hormone levels, lack of childbearing and breastfeeding, as well as environmental factors, where about 70% of, which causes cancer based on environmental factors, basically food, chemicals, radiation, and free radicals. Not forgetting lifestyle, where we have alcohol consumption, smoking, and lack of exercise being some of the reasons. And of course, hereditary, mutated genes, where we have up and down regulation of certain genes that would lead to breast cancer. The common treatments for cancer: surgery, radiation therapy, and hormonal or chemotherapy. Alternate approaches include immunotherapy, anti-angiogenic drugs, anti-inflammatory drugs, gene modulation, and bioactives. Here, we are interested to look at bioactives, where some plant-based bioactives have been shown to inhibit cancer growth. So one of the bioactives that we are interested to look at is from palm oil. And palm oil, we have got 1% of phytonutrient, and 1% of phytonutrients, and of which they are comprised of tocols, which are tocotrienols and tocopherols, and carotenes, phytosterols, squalene, lecithin, coenzyme Q10, and polyphenols. And our interest is today to look at tocols, or tocotrienols, and tocopherols. So palm tocols, or in other words called the tocotrienol-rich fraction, comprises of 70% tocotrienols and 30% alpha-tocopherol. So in this presentation, I'll be talking a little bit of TRF. And I'm sure you've seen this structure a couple of times from yesterday's presentations. So these are the basic difference between alpha-tocopherol and the tocotrienols, where we can see the major difference is the presence of the triple double bond in the phytyl chain. Tocotrienols have shown to have many health beneficial effects, and I'm sure you've heard about this yesterday: antioxidant properties, immune regulation, antibody production, growth inhibition of cancer cells, both in vivo and in vitro, now moving on to human subjects, induction of apoptosis, as well as neuroprotective properties. So coming back again to my research approach, first I'll be covering a little bit of anti-tumor activities in a cell-based system, syngeneic mouse model of breast cancer. I'll go into anti-angiogenic effects of tocotrienols, as well as some gene regulation. And I will also talk about a human study that was carried out in collaboration with the International Medical University in Kuala Lumpur. So tocotrienols and anti-cancer effects. Firstly, we started off looking into the effects of anti-cancer in mouse, human breast cancer cells as well as mouse cancer cells. And in this presentation, I'll be showing you some of the results on 41 mouse mammary cancer cells. They were cultured in 24 well plates, and we were looking, we tested using varying concentrations of tocotrienols and compared them with tocopherol isomers and carried out a growth inhibitory experiment. So here are the results that we see from the cell inhibitory experiments. This graph shows the effects of TRF as well as the individual isomers of tocotrienol. And as shown here, gamma and delta tocotrienol were most inhibitory, and whilst TRF and alpha tocopherol were quite similar, but they do also give a complete inhibition. And the tocopherols, however, the blue line here indicates TRF is compared to similar as that, and these other three lines actually indicate the individual fractions of tocopherol. So TRF is still better in terms of complete inhibition to the cell lines. Okay. So coming to the animal model, we used a syngenic mouse model, it's the bulb C mice, and they were given oral TRF supplement daily and were injected with 41 mouse mammary cancer cells and taken up for 45 days to look into the growth of the cancer cells. And at the day of after 45 days, when the tumor was just too much to bear, the animals were sacrificed, and we carried out several studies looking into the immune modulatory effects and IHC staining to look into some markers in gene and protein expression, as well as splenocyte culture to study the levels of interleukins and interferons. I would like to also say here that we also carried out a similar study on nude mice as well, looking into human breast cancer cells, but I'm not showing the data here. But basically, what we have observed, what I'm going to show you in the next couple of slides, are really what we also see in the nude mice. So this tumor growth, and we can see that it's a significant reduction of tumor incidence. The red line here indicates the TRF supplementation, and that line here is the control. And the tumor volume too were found to be significantly reduced. And when we studied the levels of lymphocytes and NK cells are natural killer cells which help to combat growth of cancer cells, we found that there's a significant increase in NK cells of the group that received tocotrienol supplementation. And interferon gamma and interleukin-4 levels were investigated, and again we found that the levels of interferon gamma was significantly increased in the TRF group. We also compared them with the group of mice that were not injected but supplemented as well, but we found that the levels were significant in the TRF that received the tumor cells. So, and there was an increase in interleukin-4. So this actually indicates that the immune system is actually undergoing the Th1 immune response compared to Th2. We went on to look at the metastasis in the lung tissue of the mice, and we found that the control animals had a larger clumps of breast cancer cells accumulated in the lung tissue compared to the control, the TRF supplemented group, which had very little metastasis in the lung tissues. And this was done on in various animals, and this is just a representative of what we saw. We went on further to look at the gene expression of what happens, in the gene regulation with tocotrienol supplementation, and this using the microarray experiment is a 3-day experiment where we have the amplifying RNA template and hybridizing it on the array, and as well as scanning and analysis. And we've actually carried out the microarray experiments on both the cells, the 41 cells, as well as in RNA extracted from the tumor tissue. And we can see that there were several number of positive genes as well as negative genes, meaning up-regulated and down-regulated genes, versus the treatment, basically the TRF, and we looked into delta tocotrienol being the most potent component. And when we went on to look at the animal, the RNA extracted from the tissue, we also found a number of genes that were up and down regulated. But what I'm going to focus on here is one important gene that we found that was both expressed highly in the in vitro as well as the in vivo model, and that is the interleukin-24. So what is interleukin-24? It's a member of the IL-10 family of cytokines that has been reported to have anti-cancer effects. And it's also known as the MDA7 tumor suppressor protein. It signals through two receptors, the IL-20 and IL-22. And it's shown to modulate the STAT1 and STAT3 pathway, MAP kinase pathway, as well as the VEGF pathway. So these two pathways actually regulate apoptosis, and VEGF pathway, it regulates the angiogenesis pathway. And it has also been known or called or referred to as the magic bullet for cancer, IL-24. So what we went on, we found this gene from our microarray experiments, and we went to investigate whether, is it really true that this gene is overexpressed in the cells? We retested using real-time RT-PCR, and we used all the isomers, but I've also done on a dose-dependent manner, which I'm not showing here. What we can see is the levels of IL-24 increased according to the potency of the components. So we know that delta tocotrienol is the most potent, followed by gamma, etc., and then the levels of IL-24 increase accordingly. When we investigated in the RNA tissues of the RNA samples extracted from tumor tissues, and we carried out a PCR IL-24, again we saw that the levels of IL-24 was increased in the TRF supplemented mice. And we have published these results because this is the first publication that showed tocotrienols could regulate interleukin-24 at the Journal of Cancer, Journal of Interferon and Cytokine Research in 2011. And since we found that IL-24 is what you call the angiogenesis, regulate angiogenesis. We also looked into whether tocotrienols regulate angiogenesis, and in the RNA samples from the mice were subjected to multiplex PCR as well as immunohistostaining to see the expression of angiogenic markers. And we found from the multiplex PCR 3 very important genes that has got are promoters of angiogenesis. So you know angiogenesis is the formation of blood vessels around the tumor cells. So we want angiogenesis to be halted so that the tumor cells do not get their supply from the blood vessels and thus causing the cells to shrink. So VEGF, through its receptors, VEGFR2 or KDR, it's known, and VEGFR1, has been shown to be down-regulated in the tumor RNA from the supplemented mice group. And this also correlates with the smaller tumors that we found in the supplemented group. The IHC results showed also that the TRF supplemented mice showed lower expression of VEGF in the tumor tissues, as well as in the lung tissues. And also we looked at one of the receptors, that's the KDR, was also shown to be reduced in the lung tissue of the mice. And we've also published this paper in Genes and Nutrition, and we went to look at, we know tocotrienols regulate angiogenetic markers, we know tocotrienols regulate IL-24, so how do the three interact within each other? And we've actually now done on 41 cells, we looked at the expression of some of the key angiogenic markers like IL-8 and VEGF and IL-24 concurrently in the cells, and we found that as the levels of IL-24 increase, there's a concurrent decrease in the angiogenic markers. So it does correlate with other observations. And yes, this is the paper that we have published on these results. And next, I would like to go through a little bit on what we have seen in a human study that is actually in collaboration with the International Medical University. So what we wanted to see whether the supplementation of tocotrienols actually enhance immune response in normal healthy subjects. So here, this is to assess the effects of tocotrienol supplementation on immune response following tetanus toxoid vaccine challenge in healthy female volunteers. So why do we have a tetanus toxoid challenge? Because prior to this, the researchers from International Medical University have found that just by supplementing tocotrienols to the healthy subject without a challenge, it doesn't do anything to the immune response. So with the with the challenge, the tetanus toxoid, and tetanus toxoid is one of the good markers that have been referred to that shows that the body has good immune tolerance. So this is the study design. We have it's a randomized, double-blinded, placebo-controlled study. 108 healthy women were chosen. And why women? Because one of the criteria of exclusion criteria was smoking, and we could hardly find male that doesn't smoke in this subject population. So we left it to just female, and they were randomly assigned based on the inclusion and exclusion criteria. Ethical approval was obtained from International Medical University, and they were assigned to control group and TRF group. The control received the placebo, and the TRF received the 400 milligrams of TRF. So the study was carried out for about 56 days, and blood samples were drawn at day 28 and day 56. A tetanus toxoid challenge was induced at day 28. And again, this was published in European Journal of Clinical Nutrition, and I'm just going to go through some of the results that we have shown in this paper. So the first one was on the anti-TT IgG. IgGs are ones used to measure the levels of response to tetanus toxoid, and we found that at day 56, after the TT challenge, we could see an increase. The white bar here, sorry, I didn't put the legend. It's here. TRF, which group that they had a higher level of IgG. The interferon gamma levels were also measured in using con-A stimulation as well as tetanus toxoid stimulation, and we found that the TRF group showed a higher response to interferon gamma in both the con-A and TT stimulated cells, and these are using PBLs, peripheral blood leukocytes. IL-24 was also looked into, and here we also find an increase in tetanus toxoid stimulated group. And we conclude from that human study that daily supplementation of 400 milligram of TRF has a beneficial role in enhancing the immune response of healthy subjects following an immunogenic challenge. So the overall finding from this study was that tocotrienols actually reduce the growth of 41 cells in a dose-dependent manner, where we find that the delta tocotrienol was more potent than gamma tocotrienol and more potent than TRF, and lastly alpha tocotrienol. And TRF supplementation enhanced the immune response by increasing levels of interferon gamma and NK cells in tumor-laden mice. Again, I would like to say that this similar observation was also observed in the nude mice. And TRF supplementation reduced metastasis, and enhance the Th1 immune response. And very interesting to show that the immune modulatory effects were associated with increased production of interleukin-24. We've also shown that TRS supplementation reduced the expression of angiogenic markers and enhanced the immune response to TT vaccine in humans. And that brings an end to my presentation this morning. I would like to acknowledge these following people: Director General of MPOB for allowing me to present here, the organizers of this conference, and as part of this presentation, as part of my PhD studies, I would like to acknowledge my supervisors, Dr. Kalanidhi, Professor Dr. Amur Radhakrishnan from IMU, and Professor Mithil Kanankuti from UITM. With that, I end my presentation. Thank you, ladies and gentlemen.
Thank you. Are there any questions from the audience? Well, if they're thinking,